All right, recording begins. This is November 5th. This is Biology 105, the year 2025. All right, so we're going to look at the syllabus real quick, at the schedule real quick. Never hurts to double check this stuff. We're here in week 11. Next week, we've got a holiday on Tuesday, right? But that doesn't impact any of your scheduled activities for this class. You know, unfortunately, or fortunately. So you get your full dose of Bio 105 next week. All right? Awesome. Yeah, very exciting stuff. So this week, we're doing meiosis. Next week, we're doing molecular genetics and inheritance. It's going to be very cool. We're going to talk about the patterns of inheritance of characteristics. It's a whole lot of stuff coming up. This week, there is no lab assignment, which is kind of a nice little break, I suppose. Yeah, people like no lab assignment weeks. And then next week's lab is pretty straightforward. You hold a bunch of laminated corn cobs, and you count the different phenotypes of the corn kernels. And then you run a chi-square analysis on it. It's going to introduce concepts which we will then really reinforce here in week 15 when we do a Hardy-Weinberg analysis on the PCR products that you will actually produce from your own DNA, saliva samples. It's going to be cool. Any questions? Oh, I meant to mention, don't forget, folks, this is week 11. Week 13, you've got another exam. I know these two are packed really tightly back to back, but you're focusing entirely on mitosis, meiosis, and then patterns of inheritance, which I consider to be probably the three most important PowerPoints, most important lectures for you moving forward in any other upper division biology courses. All this stuff will come back and needs to be understood. So I have a whole exam dedicated to it, essentially. Cool? Same length, Scantron. Any other questions? Yes?
Lab, like the pre-lab for this lab this week, was it supposed to be like almost identical to last week's lab?
Yes, because this week you are actually doing part 2 of the transformation lab.
Okay.
So last time you did like plating, right? And the pellets. And this week you're doing transformation with the plasmid. So the information's the same, it's just a two-parter lab. Sort of make sense?
It just looks like we're doing what we did last lab again.
It does, it's really similar. Same centrifugation steps and all these things. I believe that you will still be extracting plasmid DNA from a pellet, but it's slightly different. Check the handout and you'll see what I mean. It's like it's a second part of the handout.
Okay.
Yeah, is that all good?
Yeah.
Of course. And if anything weird happens with the labs, I'll have to just deal with it after it happens. But it's a good lab. You're transforming bacteria to make them be antibiotic resistant and produce green fluorescent protein. That's what you're actually doing this week. Awesome. A question?
So, since it's a part 2, are we like using the same like printout, or are we printing a new one?
If the printout that I had last week, which I think has a part 1 and then a part 2 that you didn't fill out, that's how I meant to upload it, then you can use the same printout. But hey, you know what? This is a reasonable thing for me to just check right now. Not running a lab this semester means that sometimes I forget if I've done something correctly or not, you know? Like I thought I did this. Did I? Let's find out. Let's find out on film. So it'll be immortalized, my mistake. DNA extraction, okay, lab handout. Let's look at this one compared to this one right here. Same 4-page, same, longer. So the one that's posted this week has the second half on it, but also has the first part still included in it. So you could start printing at page 5 and then combine the two documents and you'd be sitting pretty. All right? Thank you for clarifying that. Like, totally my bad. What I meant to do was upload the complete document last week, but in previous semesters, that's also caused confusion because people will be like, I printed all this. When are we doing the other part? And it's like, well, that's next week. Anyways, I did it wrong, and I accept mea culpa, mea maxima culpa. Everybody a big fan of Sweeney Todd? Yes, you are. I know you are. Okay, cool. Any other questions? Taking all questions up here. Okay. Enable editing. Boom. We're talking meiosis now. All right. We are going, I believe we finished our life cycle and we got into the stages. Enable external content for this session, please. And we got into the stages of meiosis, this slide at least, which I'm going to really, we're going to start talking about. Remember I mentioned prophase 1, metaphase 1, anaphase 1, telophase 1, prophase 2, metaphase 2, anaphase 2, telophase 2, right? Meiosis has 2 stages, okay? Meiosis 1 and meiosis 2. Both meiosis 1 and meiosis 2 result in cell division. So you go from 1 mother cell to 2 daughter cells in meiosis 1, and then each one of those daughter cells divides again in meiosis 2. Now there are 4 resulting daughter cells. They are all haploid. They all have unreplicated chromosomes, and the fate of those daughter cells differs between sperm cells and egg cells and plants and animals, and you'll learn all about that stuff in other classes. But for us, we're talking specifically about the net output, the very generalized net output of meiosis. So, broad overview with meiosis 1 and meiosis 2, and then we'll look at the stages of meiosis 1 and meiosis 2. Sound good? Awesome. In interphase, all right, we're tracking a single pair of homologous chromosomes here. The chromosomes duplicate. Now, in what phase of interphase do the chromosomes duplicate? S. I heard another S from somewhere in this region over there. S phase, that's right. Remember, S for synthesis. So these cells are going to undergo meiosis, but they're still going through interphase. Chromosomes duplicate, and now you have 2 homologous chromosomes that have been duplicated or replicated. Replicated chromosomes, duplicated chromosomes, those 2 terms are used pretty interchangeably depending on which textbook you're using, which edition of a textbook you're using, or sometimes chapter to chapter, which is just great. But duplicated, replicated, same thing. The point is that the chromosomes have undergone replication or duplication, so each sister chromatid should be more or less identical. If S phase went the way it's supposed to, each sister chromatid is identical to the other sister chromatid, all right? So here we have a homologous pair of duplicated chromosomes, sister chromatids, non-sister chromatids, all stuff that we've spoken about. Any questions about that? All right. Then meiosis 1 happens. Don't worry, we're going to zoom into what goes on in meiosis 1. It's PMAT, see? All right? But in meiosis 1, the homologous chromosomes get separated, okay? So now each of these cells is haploid because they only have 1 of each chromosome, all right? They're the duplicated form of the chromosome, but it's still 1 chromosome. Great. Then meiosis 2 will happen, and in meiosis 2, sister chromatids will separate. And when the sister chromatids separate, we end up with non-duplicated or unduplicated or non-replicated or unreplicated chromosomes. So the chromosome number did not change during meiosis 2. You went from a cell that, say, had 23 duplicated chromosomes to now having 23 unduplicated chromosomes. But you went from 23 chromosomes to 23 chromosomes. I know I'm being a real stickler about, like, this is a chromosome and this is a chromosome, but it's a standard stumbling block for people, and I really want to make sure we got it. Yeah? In meiosis 2, so here's the duplicated one. And remember in mitosis when the kinetic cores formed and these things got yanked apart? I did this, I was standing in front of another one of me and I went like, whoa, exactly. That's gonna happen here in meiosis 2. So meiosis 2 is very similar to mitosis in that the sister chromatids get yanked apart by microtubules. Yeah.
And that's it. That's the only thing that makes them unduplicated.
Yeah, exactly. Yep. Cool. Question.
So meiosis one, let's say there's 23 chromosomes.
Totally.
So there's 23 chromosomes, and then meiosis two, there's 23 chromosomes.
Exactly.
Is there
Or,
double the
sorry, I want to make sure that I just want to make sure that we're talking about the same thing. After meiosis 1, there are 23 chromosomes. And then after meiosis 2, there are still 23 chromosomes. Is that right? Okay.
But there's double the amount of sister chromatids.
There are no sister chromatids following meiosis 2.
So how does it go from 23 to 23?
They get, so this is a chromosome, this is a chromosome, these are chromosomes. So if this is in one cell, and then you make 2 cells and each one gets one of these, you went from 23 of these to 46 of these, but 23 went to one cell and 23 went to the other cell. So the net number of it all comes down to how we define a chromosome. Because we're defining this as a chromosome and we're also defining this as a chromosome.
But separately as chromosomes.
Yes, that's a chromosome, that's a chromosome.
Okay, so
And that's a chromosome.
there's 46 after in 23 in each cell.
There are 23 chromosomes before meiosis 1, there are 46 chromosomes. We're talking about humans, of course. So in humans, there are 46 chromosomes. Check this out, right? G1 of interphase, 46 chromosomes in a human cell, because there's 2 copies of each of the 23 chromosomes. S phase happens and there's DNA synthesis. After S phase, you've got 46 total chromosomes. They're duplicated now. So there's twice the amount of DNA, but there's still the same number of chromosomes, because now they're just in the duplicated form. Then, following meiosis 1, you actually reduce the number of chromosomes, okay?
By breaking it into different cells, right?
Yes. By taking each homologous pair and putting it into a different cell. Yep. So then you're down to 23 chromosomes in each of these. But then, after meiosis 2, you've just gone from duplicated to unduplicated chromosomes. So the number of chromosomes has not actually decreased, even though the amount of DNA has decreased. Yes?
So is it fair to say that with duplicated chromosomes, it does not matter that they're duplicated because the information within them is identical?
That's why we still call it the same chromosome. Exactly. I'm going to repeat that for the recording and for anybody who didn't hear it. We don't consider this as 2 separate chromosomes, right? Because each sister chromatid's identical. You have produced something where each arm of the chromosome's the same. So we don't consider that as 2 separate chromosomes because they're the same. They're also being moved about as a unit until that centromere gets degraded. So, it's a lot to think about. I want to draw something for you.
But just to clarify,
Please.
in meiosis 2,
Yeah.
23 chromosomes in each cell. Once the sister chromatids separate.
Yeah, before they separated, there was also 23.
Yes.
Yeah.
Once they separate, there's 23 in each cell with a total sum of 46. If you combine those cells.
If you combine the cells, but don't.
But don't. Yeah. No.
Yeah.
I'm just trying to make sense of it.
That's exactly what we're trying to do. That's why we're in the dang old class, so I'm 100% there with you. So let's go ahead and annotate a little cell cycle of a cell that's going through meiosis. So we're going to go with human. Oh. I'm going to keep that up here. I'm going to do this here. That looks good. Let me zoom in so the image looks better. Okay, so we've got a cell that's undergoing meiosis. It's got a G1 phase, an S phase, a G2 phase, an M1, a meiosis 1, and a meiosis 2 phase. All right, this is a human cell, so the diploid number is 46. There are 23 distinct chromosomes. Cool so far? I'm going to get a different pen out of my bag. There we go. Human. All right. 23 unique ohms, 2 copies of each. Okay? That's like the situation. So in G1, you have 46 total chromosomes. All right. 2 copies in homologous pairs. Homologous pairs. All right. In S phase, you go to 46 total chromosomes duplicated. All right, that's the difference, is you've gone from the chromosomes are like this to the chromosomes are like this. In G2, you've got the same as S, all right, 46 total, duplicated. All right, in M1, you've got 23 total, or I'm going to say produces 23 total duplicated, because the homologs have separated. Oh, sorry, I said hot dogs there. I think I might have spelled separate wrong, but that's fine. In M2, that produces 23 total. All right, and this is actually going to leave. Okay. Produces 23 total, okay, unduplicated. All right, and you end up with 4 daughter cells, each of which has 23 total chromosomes unduplicated. Question?
So when you say produces, that's like on top of the previous one.
What I mean is that the process of meiosis 1 produces 23. Not that when the cell is in meiosis 1, it necessarily has this. So I could also, instead of saying produces, because they start with the same food, the same like stock. I was going to call it a feed stock. They start with the same material. But here at the end of meiosis 1, once meiosis 1 has completed and the cell enters meiosis 2, there are 23 total chromosomes duplicated because the homologs separated. And then here at the, perfect, I'll just do it like this. And then at the end of meiosis 2, you get these 4 cells where there are 23 total chromosomes and they are unduplicated. All of the DNA generation happened in the S phase. So there's no new DNA being created here, 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 or here. You generate your total quantity of DNA in the S phase. And that's what you're going to have as you enter the M phase. Yes?
How many cells would be the 46 total duplicated chromosomes before you hit meiosis 1?
Oh, this is just a cell-by-cell basis. So
it's one cell.
Yeah.
46 total duplicated chromosomes in one cell.
Inside of that cell, yeah.
Okay. And then you split them up. Okay, you get 4 with 4 daughter cells for variability. You can keep this around.
Yeah, these are your gametes now that are going to be used in fertilization. Yeah, does that kind of make sense?
Yeah.
Awesome. Awesome. Okay, so I'm going to add a last little thing here. DNA quantity increases. Okay, because DNA quantity increases only during the actual S phase of the cell cycle. All right? And that's true with mitosis, that's true with meiosis, and that's definitely 100% on your next exam. Okay? When does new DNA get synthesized? And I'll ask the question in a trickier way than that, but you know, if you remember this, you'll be able to answer that question. Okay. Now, I'm tired of looking at my disgusting handwriting. You know, I'm going to blame it on the fact that I'm writing like this, but really, it's because, you know, once you get a PhD, they smash your hands. So be happy that it looks as good as it does. All right, here we go. Let's keep on crushing material. This is a very simple view. Sometimes when we get to the more thorough view of meiosis, it actually like makes more sense because there's actually enough detail to explain what's going on. So let's just continue by looking at meiosis 1. All right? Meiosis 1, the process which separates the homologous chromosomes. All right? Cool. So here we are in prophase 1. All of the chromosomes have been duplicated. The nucleus degrades. The spindle microtubules extend, just like in prophase of mitosis. Why does the nucleus need to degrade? So that the spindle microtubules can attach, or that the kinetic core microtubules can attach and move the chromosomes around. Now, during this stage of prophase, right, or actually, maybe kind of prior to this, but there's this point where the chromosomes align with one another, the homologous chromosomes align, and they do this process of crossing over, where material is exchanged between the homologous chromosome pairs. This generates what we call recombinant chromosomes. And I drew something about that last time. I tried to indicate that process here, where in meiosis 1, there's some crossing over, where materials exchanged between homologous chromosomes, which generates new recombinant chromosomes that have some maternal and some paternal DNA. All right? But this crossing over process is really important for generating additional variability in sexual reproduction. So anyways, we'll come back to it. We'll talk more about it, but let's just look at prophase, metaphase, anaphase, telophase, and cytokinesis. So when we're talking about meiosis, we usually just don't even talk about pro-metaphase, which is pretty annoying, but you know, we'll make it work. So prophase, the duplicated homologous chromosomes, they're all sort of stuck together because they're undergoing this process of synapsis and crossing over, all right? The kinetic core microtubules extend, the spindle microtubules, they attach, okay, to the chromosomes, the homologous pairs of chromosomes, which are all joined together to one another. So it's kind of similar to sister chromatids being joined at a centromere, all right? So they all get aligned at this imaginary metaphase plate. But now, look, it's every homologous chromosome pair. So when the chromosomes get yanked apart in anaphase, meiosis 1 is separating homologous chromosomes so that each daughter cell gets one of each chromosome. So you go from a diploid cell here with 2 copies of every chromosome to 2 haploid cells here with 1 copy of each chromosome. And additionally, there's been some recombination where material has been exchanged between the homologous chromosomes. So some of the sperm chromosome 1 and some of the egg chromosome 1 got swapped, all right? Which is how I like to refer to paternal and maternal DNA, because I think it's a little more accurate, you know? Yeah.
So the crossing over process only happens like around prophase?
Yes, the crossing over process occurs in meiosis 1 in prophase 1. That's when you've got the crossing over process. It involves these proteins, you know, everything involves proteins, and they form this thing called a synaptonemal complex, which you won't be tested on. Yes?
Out of curiosity, with like prophase to cytokinesis, could there be an exam question where you have to like identify, oh, this is prophase or this is metaphase?
Yeah, there could be like a picture of the cells, and I'd be like, hey, the cell indicated by the letter C, is it at which phase? There also could be a question about like, in which stage does a cell is at anaphase 2, how many sister chromatids are present? Something like that, you know. Yeah. And the answer there would be 0, because at anaphase 2, the sister chromatids got separated. But I'll show you more about that in a second. So crossing over, synapsis and crossing over, right? Homologous chromosomes align. They're very similar to one another. and there's these enzymes that are involved, these things lay up, and then they get snapped together, and it crosses over. There's all these names for this process. These cohesin proteins are cohesive. They attach the two things together. None of that's important for our understanding. You simply need to know that crossing over occurs at the beginning of meiosis 1. If you know that, then you are good to go, okay? Crossing over. It occurs between non-sister chromatids of homologous chromosomes. So here's a little diagram of it. The homologous chromosomes align, the chromosomes cross over, and now you get these recombinant chromosomes where this was capital A, capital B, capital C, and now it's capital A, capital B, lowercase c. You see, this is one of those things where I can say it, but I think that looking at it makes a lot of sense. So I want you to just look at this diagram for a second and read that text, and I'm just going to be quiet for like 15 seconds while you let the information actually hit your eyes. All right. It's wild how long 15 seconds is. Okay, so what do you think? Questions about crossing over? Questions about what makes a recombinant chromosome versus a non-recombinant chromosome? Yes?
I just want to make 100% sure this occurs
Please.
at the very beginning of meiosis.
At the very beginning of meiosis 1, these synaptonemal complexes form, the chromosomes are all attached, and that's going to make it really easy for them to all be aligned at metaphase 1. And then the homologous chromosomes will be separated at anaphase 1. So the synaptonemal complex, or crossing over, is occurring at prophase 1, and it's done at anaphase 1. And that's the only time crossing over is occurring in metaphase. Awesome question. Awesome. Yes?
I was wondering if you can give a definition of the
Recombinant.
Yeah.
No problem. Yeah, the recombinant chromosomes. Absolutely. So
It's mixed with DNA, right? Like it has the crossing over stuff. I don't know what would be a good definition of that.
no problem. It's a mix of paternal and maternal DNA.
Okay.
Yep. So this chromosome was entirely paternal. This chromosome was entirely maternal. And after crossing over, you've got this chromosome that is a mix of maternal and paternal DNA, and this chromosome that's a mix of maternal and paternal DNA. Meaning that this chromosome here came entirely from a sperm cell. But this chromosome, and this chromosome here came entirely from an egg cell. But this chromosome here now has sperm and egg DNA in it, as does this chromosome. Does that make sense?
Yeah.
Too easy. Who's next? Who's got questions for me? Awesome. So there's meiosis 1. We come back to it. I bring it up again. And now we've discussed the crossing over piece, so we can just focus on homologous chromosomes aligned at the metaphase plate, and then they get yanked apart. Cool. Awesome. So then there's a telophase at a cytokinesis stage where new nuclei sort of form. The cytoplasm gets separated, there's a cleavage furrow, and you get 2 daughter cells that are now haploid. So from diploid to haploid. Then, oh yeah, please. I will. All right. Meiosis 2. In meiosis 2, the sister chromatids get separated. It's just like in mitosis, okay? Except there's no homologous chromosome pairs. But otherwise, the process is the same, right? It's all duplicated chromosomes. They all align at the metaphase plate. Kinetochores form. Anaphase happens. Sister chromatids get separated. And now you've gone from, in a human, 23 duplicated chromosomes to 23 unduplicated chromosomes. This process of meiosis 2 does not reduce ploidy, and it does not reduce chromosome number, okay? It just goes from duplicated to unduplicated chromosomes, all right? And that's going to generate 4 total daughter cells after meiosis 1 and meiosis 2 have completed. And these daughter cells will not be identical to one another, and they will not be identical to the mother cell that produced them. All right? We cool so far? Please.
Just to reiterate, 4 haploid daughter cells, non-identical.
Yes, exactly. Mitosis produces 2 identical daughter cells. Meiosis produces 4 non-identical haploid daughter cells. Of course. And just to be a jerk, some organisms aren't diploid, so meiosis wouldn't make haploid cells out of them, but let's not worry about that. Let's focus on humans, where we're diploid. Let's focus on diploid organisms. Yeah. Good. Additional questions there? Yes, please. It is only in prophase 1. Yep. So that's where we have the crossing over, and that's where the recombinant chromosomes get generated, is in prophase 1 of meiosis 1. Awesome. And then remember, this can get really tricky, folks. We're looking at an example here with 3 chromosomes, but remember, there's 23 in a human. There's more in other organisms, okay? So there's a lot of recombination occurring. There's a lot of random assortment of which chromosome's gonna go where here in anaphase 1. So imagine this, but with 20 additional chromosomes. And you can see how much variability could be generated in between the different gametes. We good? I'll come back to that again, though. Yes?
Just can you repeat the piece about the 2 daughter cells and then the 4? Mitosis does 2 daughter cells.
Yeah, absolutely. So let me just write it down. It seems like a good important rule of thumb that people would like to just have. Main projector, document camera. Mitosis, meiosis. Mitosis, 2 identical daughter cells. Meiosis, 4 non-identical haploid daughter cells. I'm going to put the word diploid here. Cool?
They're not identical because of the crossing over part that happens.
Not only that, but they also have been reduced in their ploidy, right? So you had the crossing over, boom, boom, and boom, boom, and the reduction in the ploidy number. So reduction in ploidy. So immediately, these first daughter cells aren't identical to the initial cell because they only have 1 copy of each chromosome. And then these 4 daughter cells aren't identical to one another because not only has there been crossing over, but there's been random provisioning or random assortment determining, like, as a sperm, I have paternal chromosome 1, recombinant chromosome 2, maternal chromosome 3, maternal chromosome 4. Chromosomes 5 through 7 are all paternal chromosomes. The chromosomes 8 through 10 are maternal chromosomes that have undergone recombination, and they are 10 to 18% paternal DNA. And then another gamete next to me has a completely different arrangement of chromosomes and what they've received. Does that sort of make sense? So that adds to all the variability in the gametes. Cool. Alrighty. Good. So here's the key thing here is anaphase 1 homologous chromosomes get separated, anaphase 2 sister chromatids get separated. Everybody cool with that? Awesome. A comparison of mitosis and meiosis. Oops. I'll put this slide back on for anybody watching the recording. This is what that slide was, where I said the key thing is anaphase 1 and anaphase 2, what you're separating in each one. Here's a slide that compares mitosis to meiosis. All right. Yeah. We saw mitosis. Look, you got the duplicated chromosomes, 2 copies of each chromosome. The daughter cells get generated, and they're no longer duplicated, but you still have 2 copies of chromosome 1, 2 copies of chromosome 2, etc. So there's mitosis on one side. Here's meiosis on the other side. both of them start with the same diploid number of 6 total chromosomes, meaning 3 distinct chromosomes, but 2 copies of each. Okay, so both cells start the same, but in meiosis, there's a crossing over event at the beginning, and then in metaphase 2, or metaphase 1, homologous chromosomes are separated, and then in meiosis 2, sister chromatids are separated.
So crossover, just to double check, only happens in meiosis.
Only in meiosis, only in meiosis 1.
Only in meiosis, only in meiosis. Okay.
Yep, perfect. Good, I'm just scanning the room. Yes,
In pro or metaphase?
prophase. Yep, yep. And it's like, oh, technically it can begin at the end of G2, but where we determine is G2 and prophase and when one starts at the other is kind of an arbitrary distinction. It's like the chromosomes condense in prophase and then crossing over occurs, but it's happening at the same time. So making them sequential is tricky. But yeah, we'll just say prophase 1, crossing over happens. Awesome. Additional stuff. Okay, so there's people like the comparison of the two a lot, so I like this table. Now what changed? Did anything change? Did I change something there? Nope, I just have 2 copies of the same slide. Good, good to have. Genetic variability, genetic variation. Meiosis exists, okay, to generate more variability. Sexual reproduction exists to generate a population with a lot of variability in it, okay? A population with more variability, it's more likely that some of those genotypes, some of the combinations, some of the varieties of offspring are going to be better suited to their environment, or that they'll be better suited to the environment as conditions change, okay? So this whole thing exists to generate variability, and variability in gametes is generated by two processes. The crossing over event that we've already talked about quite a bit, and then actual alignments of the chromosomes at metaphase 1, which I'll explain in a little more detail. So originally, all of the genetic variation occurred due to mutations. These mutations are usually the result of errors in DNA replication in S phase. And sometimes you get an offspring that happened to have a different sequence of nucleotides that happened to make a protein that was a little better than the previous version. And so then natural selection encourages more of those individuals to be, produced or to survive. And everything's reproducing asexually. Everything's reproducing via mitosis. So mutation's the only way to generate variability, okay? Mutations create different versions of genes called alleles. Is everybody pretty comfortable with that allele conversation? I've brought it up at least twice. Different versions of genes are called alleles. Sometimes there's a version of a gene that is a non-functioning version, and that's like a non-functional allele. It's still a version of the gene, it just doesn't work, all right? So because we're diploid, we could have one allele that functions and works and makes what it's supposed to make, and then another allele that's non-functioning, but that one copy of the functioning allele is good enough to allow us to survive and thrive and do all the things that we need to do, okay? Cool? Awesome, I love it. Reshuffling of alleles generate variability, very good. 3, okay, yeah. The third mechanism here is a boring one for generating variation in offspring. It's just like, you don't know which sperm's gonna fertilize the egg, so it's random. Fine, boring. But independent assortment of chromosomes and crossing over are 2 things that are a little trickier to understand. Crossing over, we've already spoken about, right? It generates completely new, novel, recombinant chromosomes that are a combination of maternal and paternal DNA. They haven't existed before, this specific chromosome structure. So that generates variability. We're cool with crossing over? I know I've already asked. Okay. Now, this one right here, independent assortment, it's I don't know what it is about it, but it gives people such a hard time. It is just, it's People just don't get it. And I've tried explaining it in a bunch of different ways, and I don't know. I'm at a loss, you know? It's just complicated. It is complicated, but once you get it, it's like, oh, obviously. It's one of those, and those are frustrating. Trust me, you can get there. Check it out. The way that the homologous chromosomes align at metaphase 1 is random. What I mean is that you could potentially have all paternal chromosomes on the top and all maternal chromosomes on the bottom, or each chromosome pair, each homologous chromosome pair, can be flipped with maternal on the top, paternal on the bottom. The next one could be maternal, paternal, 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 maternal, 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 paternal, paternal, maternal. Something like that. You see what I mean? So there's a bunch of different possibilities. When you've got 23 different chromosomes, there's a ton of different possible arrangements. Let's look right here at these two arrangements, okay? At metaphase 1, in this example, it's paternal, paternal on the left, maternal, maternal on the right. In this example here, it's paternal, maternal, and then maternal, paternal, all right? In metaphase 2, after these homologous chromosomes get separated, that means that this cell is all paternal for these two chromosomes, and this cell is all maternal for these two chromosomes. So after they divide again, you're going to get a gamete that's all maternal for these two chromosomes, and you'll get two gametes here that are all paternal for these two chromosomes. But if the chromosomes were aligned differently at metaphase 1, then the daughter cells of meiosis 1, okay, would be a mix of maternal and paternal DNA. So these are two potential arrangements of chromosomes, two potential situations, okay? And each one produces entirely different gametes. If there are 23 pairs, then the number of possible combinations is 2 raised to the 23rd power, which is a crazy big number. So it generates a lot of potential arrangements of chromosomes in the daughter cells. So, first, orient. These are just 2 equally probable arrangements of chromosomes at metaphase 1. These are just 2 possible situations. We're about to begin our discussions where we're talking a lot about probabilities, okay? And we're talking about probabilities. It's important to know that these aren't 2 cells, all right? These are 2 possible arrangements of chromosomes, okay, at meiosis 1. Yes, please.
So it's kind of like making a sandwich type of deal. It's like you can either have, like, do you want peanut butter and jelly, or do you want, like, ham and cheese, kind of? Is it like that where it's like, there's only, like, so many outcomes you can do?
There's only so many outcomes, but it's like, it's more like you've got peanut butter and jelly you inherited from the paternal side, and you've got ham and cheese that you inherited from the maternal side. And you can create a gamete here that's peanut butter, jelly, ham and cheese. Solid. But it's completely random which ingredients you receive. So these gametes are peanut butter, ham, jelly, cheese. Is that what you were going for there? Yeah, that's a good analogy. I like it a lot. I'm going to give you 1.7 John points, rounds up to 2. Put them in your pocket. They last until the end of the day. Very good. So yeah, you've only got so many ingredients available here, but which combination of ingredients you receive is all dependent on how the chromosomes aligned at metaphase 1. All right? There are actually 4 potential arrangements when you have just these 2 chromosomes. All right? You could have these top ones flipped and these bottom ones, or these bottom ones flipped, and then et cetera, et cetera. But what that means is that each gamete can be entirely different from the other gametes. Add on top of it that there's been recombination going on, and now you get recombinant chromosomes thrown into the mix, and you've got 23 chromosomes, remember? So there's a lot of different options of how this can happen. So the 2 mechanisms, and then the third one's random fertilization, it's boring, but the 2 primary mechanisms that generate variability are the random alignment of chromosomes at metaphase 1, and then crossing over, okay? And then which sperm's gonna fertilize the egg? You don't know which one's gonna hit, all right? So that's the random fertilization piece. Now, I've got the summary, I've said this a million times. I've said this a dozen times. Let's be reasonable, okay? Sexual reproduction creates variable populations. That's the purpose, okay, of it. Here's a summary table of mitosis versus meiosis. We'll come back to this on Friday. In your lab this week, you're dealing with genetics, okay? You're dealing with dominant and recessive traits, something that we haven't necessarily, but we have not spoken about in lecture yet. So really make sure that you watch that pre-lab video and that you listen to the dang, wait, no, that's next week. Never mind, that's next week. We'll have spoken about it. You're good, but still watch the video, read the thing. Have a great day. I'll see you all on a Friday. Goodbye, everybody. What's that?